welcome to the Mobilize and Restore webinar series, and thanks to those of you who continued on from the research talk. In this portion of the webinar, Allison Clouthier will lead a tutorial for mapping soft tissue attachment sites across bone shapes generated from a statistical shape model. And again, we'll take more questions at the end of the tutorial and use that Q&A panel. Feel free to enter any questions as she's talking. So with that, Allison, go ahead and start the tutorial.
Okay, thank you. Okay, there we go. So I think you should all have the link to this Google Colab, so you'll be able to go through it on your own as well, but I'll kind of walk through the steps and what we did in this paper and kind of see the end result as well. So I have some links to different resources related to this here. But basically what we're trying to do is see what the effect of some of these geometry features are on the biomechanics. So we use this statistical shape model to find those shape features and then do some simulations to see what the effect of those are. So this is kind of a preview like what Aaron was just showing, where we were looking at how this change in the trochlear groove geometry impacted some of the kinematics and ligament forces and things like that. So we'll kind of walk through some of the steps of creating the morphable model in this tutorial. So I'll skip over that's just installing some of the packages, but this is the overview of the workflow. So starting from basically having your segmented MRIs or whatever images you have, creating the morphable model, running simulations, and then seeing the impact on the biomechanics. So the tutorial focuses on these highlighted cells here related to creating the statistical shape model and the morphable model. So we'll go through some of those steps that Aaron was talking about. So the first thing that we're doing here is just importing the reference mesh because we need a reference geometry for the shape model and a reference model that has all the ligament and wrapping surface and muscle attachment information from your model. So that's basically the first step here is just to import the reference geometries and then we'll visualize them. So here, since I had bone and cartilage, I've created this structure to kind of keep track of those. So depending on what kind of data you have, you might want to do something like that. Or if it's all just bones, maybe you can just create a list of the bodies and you don't need to have those separate. But here's our reference mesh. So we'd want to make sure that it looks like a good quality mesh, and this one's nice and smooth, and it's the average of the participants that we had in this study. So it's kind of representative of that. So that's our reference mesh. And then the next step here is importing all the meshes we want to use to create the statistical shape model. So here, and it's on the GitHub page, that there's a link to it at the start of this Colab notebook where it has some functions and the data. So there's 100 knees there. They're synthetically generated from a statistical shape model based on a previous statistical shape model. So you can import all 100, which might take a few minutes, or if you want to go more quickly, you can reduce the number here. But I've imported all 100, and then I've just plotted all the femurs just so you can see kind of what we're dealing with. So this is all the femurs overlapping. So you can see you have a whole bunch of femurs and they're not quite aligned. And we also have different shapes, obviously. So the next step, like Aaron mentioned, that's really important is getting that node correspondence or point-to-point correspondence. So this is what she was talking about with those points on the face, trying to make sure we have the same node number on the same anatomical locations. So in this data set, it already has node correspondence, so that should already be done and we don't need to worry about it. But if you were creating your own, it would be a really important step. And I have a list of some examples of different code that's available for doing that, or like the ShapeWorks, I think is more of a GUI that you can use to do that. So there's lots of different options. Sometimes depending on what you're doing, one might work better than the other, but you can probably just find whichever implementation works best for you and use that. So in this example, like I said, because it was synthetic data, it already has node correspondence, but we can just confirm to try and get some reassurance that that's actually working. So here I plotted 5 of the femurs And I just picked a few different node IDs kind of at random and plotted those points. So they should all be on the same anatomical location on the same knees. And that is what we're seeing. So like this yellow point is on the same spot on those different knees. Same with this purple point, it's just behind the epicondyle. So it does seem to be on the same location. So that's kind of giving some reassurance that that correspondence is working. If we wanted to be really sure, we could check more points or color the whole mesh just to confirm that. But since we were pretty sure we already had it, this gives us good confirmation that that's done properly. So our next step, like Erin mentioned, is to align and scale the meshes. So we need to line them all up, translate and rotate them so they're all aligned. And in this case, we're also scaling them. That's optional depending on if you want your shape model to also capture the overall size, you can not scale them. Then your first principal component will probably be the overall size. So here we weren't interested in size. So that's why in this Procrustes function here, I've set scale to true. So it's going to scale them. And we're basically, since we have this point-to-point correspondence, use this Procrustes function that's just going to minimize the distance between the matching points. So if we have these two patellas, it's just lining up the corresponding points the best way it can, basically. So that's what I'm doing here, running this Procrustes to align everything to our reference mesh. And for the cartilage, I'm applying the same transformation that I did for the bone, just so that it stays with the bone it's supposed to be attached to. So here I'm showing the femurs after the Procrustes alignment. So we can see now they're all nicely aligned. They're all the same size. There's obviously some variation in the shape between them, but they're all lined up, which is what we want. Now we could check the patellas as well, just to make sure that that's also good. And we see those are lined up too. So now that we have all our meshes prepared and aligned, we're ready to create the statistical shape model. So here we're using principal component analysis. So to prepare the data for that, basically we need to create a matrix where each knee, in this case, is going to be one row in the matrix, and then we're putting the X, Y, Z coordinates of all the bodies along the columns. So we're creating this basically huge X matrix, and that's what I've done here is just compile all those node coordinates into a matrix that we can use for the principal component analysis. So here I printed out, that's our X matrix of all the coordinates. It has dimensions 100 by 97,101. So 100 for the 100 knees. And then these are all the point coordinates that we had. So once we have that, we can just apply principal component analysis on that matrix. So I saw there was somebody asking a question about how accessible it is. So there's PCA functions in Python and MATLAB. So it's not too difficult to find. So we have a function for that in Python that we can just use. You can specify how many components you want. So I've just put it to 9 here, because like Erin was talking about, the first ones are going to be explaining more of the variability. So those are kind of the ones we're more interested in looking at usually. But you could go all the way up to 99 in this case if you have the full 100 meshes. So you can have up to n-1 components that you extract. And what we're going to get out of this are the principal components, so our shape features. So I've called that U here, and the scores for each shape feature. So each knee is getting a score for saying how well that shape feature is represented in that knee. So we're getting those two things out of this shape model. So we can then visualize those shape features, like some of those animations Erin was showing, to see what they actually represent. So we're basically reconstructing from a low to a high score for that shape feature to try and see what it represents. So in this case, here I've set it to PC 0. So since Python starts counting at 0, this is our first principal component. And I just have a function to split the knee back up into the different bodies because I had compiled them all for the shape model. And so we're going to look at -3, 0, and 3 standard deviations of this principal component to try and see a low score, the mean, and a high score. We'll reconstruct the mesh for each of those scores. And that's what's plotted here. So the blue is the low score, the red is the high score, and we can kind of look at what's different between them, see what this shape feature is representing. So the main thing that's being captured here is how high up this cartilage is coming on the femur. So you can see the higher score means the cartilage was up higher. For these lower scores, we have a lower cartilage. And you could look and try and see what other things might be captured by this as well. There's usually a bit of everything. And if we want to see a different shape feature, we could look at principal component 2, the second principal component. So for this one, this is one of the ones that I had looked at in the paper. So it was kind of capturing the depth of this trochlear groove. So here, in this case, the low score, we have a sort of a deeper groove, and this one, a higher groove. And the condyles are moving further apart or closer together as well with this shape feature. So that lets us kind of understand what that is actually capturing, what it actually represents. So now we have our statistical shape model, and we want to kind of make a morphable model out of that. So we had our reference model that had all the ligaments and wrapping surfaces. So we want to update those as well based on the new geometry. So we'll start with the ligaments. And what I did here is I have this JSON file that has a bunch of data about all the ligaments from the model. So it's based on the Lenhart knee model. I have a reference to that in this paper. And it's basically, I just extracted the information from the .osim file. So where it has all the ligament information, the coordinates of the attachment, and what body it's attached to, and the slack length and things like that. I just pulled that out and put it in this JSON file to make it easier to work with. So if you had your own model, you could do that as well, just to keep that a more accessible for your code rather than trying to pull it out of the OSIM file every time. So to start here, I'm just finding, looking at what we have on the reference mesh. So I'm looking at the ACL, so I'm just finding all the ACL attachment points, and we're going to plot it on the reference mesh just to see what that looks like on our reference model. So this is all the fibers of the ACL and where they're attached on the reference model. So if we just take those same points and try and plot them on a new mesh, we can kind of see what that looks like. So here I've created a new mesh. So this is 3 standard deviations of the second principal component. So I've reconstructed that mesh, this new mesh. And then if I just take those same attachment points and plot them here, we can kind of see what it looks like. And if you start looking closely, if I try and zoom in here, you might notice some of these attachment points, they're not actually attached to the bone, they're just floating in space. So since this shape is a bit different, these attachment points aren't really working anymore. So they're slightly off. And if you had a very different shape, they might be a lot further off. So we want to have those attached to the correct location on the new shape. So what we can do to do that is go back to our reference mesh and find, since we have the same node numbers for all the meshes, find what node ID each ligament is attached to. So that's what I've done here. So we're back to the reference mesh. I've just found the node numbers for what node is closest to the attachment point of all those points on the femur and the tibia. And then going back to our new mesh, instead of just keeping the same coordinates, we say if the ligament's supposed to be attached to node 100, what's node 100 on our new mesh? The coordinates for that, attach it there. So doing that, we get the ligament actually attached to the bone where it's supposed to be. So now that's updated based on our new shape. So the other thing we would want to update are the wrapping surfaces. So in these OpenSim models, they have the ellipses and cylinders and things that the ligaments and muscles are wrapping around so they don't penetrate the bones. So if we have a new geometry, we would want to update those as well so they match the geometry. So I'm showing an example for one of the wrapping surfaces here for the capsule. So I have, again, a JSON file called fitpoints.json. So this is on the GitHub as well. So in this one, I've just identified some nodes that can be used to define the shape of each different wrapping surface. So in this case, basically I had these red points are the nodes that were identified for this wrapping surface, and then I just fit a least squares cylinder to those points, and that defines our new capsule surface. So it's kind of different for each of the wrapping surfaces. Some of them are a best fit shape like this. Some of them, the dimensions are defined based on certain points. So it's a little bit different depending on what worked best for each surface. But doing this, then we get a cylinder that nicely matches the actual geometry that we have for this new model. So then we have, I'm just showing, basically updating everything for our new geometry. So it's that same new geometry for PC2. And I have there's a function called update geometry that's on the GitHub page, and it will just update all the wrapping surfaces and ligaments together. So if we create our new geometry, update everything, basically this is what we get. So we have all these wrapping surfaces that now match the new geometry, all our ligaments that are attached in the correct places on the bones. So that's updated all those things based on our new geometry. So that's kind of the end of the interactive part, but then just to show some of the results we found after that, once we had that morphable model, we could put it into the OpenSim model. So we're using the OpenSim Jam or COMAK framework. So I have some links to that in here as well. But basically how this works is it's an add-in for OpenSim that allows you to have this 12 degree of freedom knee. So the knee flexion angle is going to be defined based on the inverse kinematics, so whatever you measured using motion capture. And then the other degrees of freedom, so everything for the patella, the tibia rotations and translations other than flexion, are all optimized at the same time as the muscle forces. So that means that if we change the geometry, those secondary kinematics can change as well as the ligament forces and muscle forces and cartilage contact pressure. So it creates a nice framework where we can change the geometry when having the same overall sort of gross motion and see how that impacts the secondary kinematics and forces. And like I said, there are links in here if you want to find out more about COMAK. There's a SimTK page for that and things like that. So once we are able to basically put our morphable model into this full body model, we can run the simulations for the same walking trial. And to kind of follow up on one of the questions too, one of the other challenging things about incorporating this is how you want to align your knee model into the full body model. So we used the approach of following an automated procedure for creating the coordinate system for the knee and then just aligning that with the OpenSim model. But depending on what you're doing, you might want to align that a different way. So some of the results we found, this is for that second principal component that was really capturing this trochlear groove depth. So we made different instances from the shallow to the deep trochlear groove. So the blue ones here are the shallower groove and the red is the deeper groove. And we were able to look at what impact that had. So we saw that with the shallower groove, the patella was tilted more laterally. It was translated more laterally. And then we could look at the ligament forces as well. So we saw that that medial patellofemoral ligament had a lot more force in the shallow trochlear groove knee. So it seems like that ligament is doing a lot more work to try and restrain the patella when you don't have that constraint from this deep trochlear groove. So it allows us to look at kind of what the impact of these shape features are and how they could possibly play into different pathologies or things like that. And then we can also look at the cartilage contact pressure. So this is for the gait trial. On the left is the shallow groove and the right is the deep groove. And we saw that the contact pressure was greater and more concentrated in the shallow trochlear groove. And then just one of the other shape features we looked at was PC3. So this one was more related to the tibial plateau. So we had these blue ones here were a larger medial tibial plateau that was more flat, and the high scores, or the red ones, had kind of a sloped plateau in the frontal plane and a smaller tibial plateau. And we saw that that impacted the internal rotation of the tibia, the anterior translation, and then also the ACL force. So again, just giving this way for us to, once we have identified these shape features, see what impact they can actually have on the joint function. And then I've just included some more resources here, so links to the paper, COMAK, and also what Aaron had already talked about, our ISB workshop and those resources. Yeah, so I'm happy to take any questions that anyone has about that.
Awesome. Great. Thanks, Allison. And now we'll go ahead and switch back to the Q&A. And we'll try to focus on some of the ones related to the tutorial, and then we can loop back to some of those ones we had earlier. One of the first questions during the tutorials from Scott Stanley was how many 3D surfaces do you need, or have you found to need to build a statistical shape model, and how does that relate to the variation in shapes? I guess does it depend on the research question, or maybe speak to some of the experience you had there?
Yeah, so it does kind of depend on what you're doing, what the bones are that you're looking at. There's not really a set number. So in this paper, we had 14 knees included, 14 healthy knees. So that's probably on the lower end. It's better, basically more is better, then you're getting a better representation of the actual variability you have. But technically, I think you could do it with 3, but it just probably wouldn't be, it's not going to give you anything too useful because it's only looking at variation in those 3 people. So more is better. And there are some methods that people have proposed for trying to figure out, like, are your modes changing if you add more meshes? So you can kind of test it. But in general, it's kind of one of those things where just the more you can get is probably going to be better.
Yep, definitely. The next question is from Clarissa Lees. So when combining the multiple bone and cartilage models to run PCA on, is the PCA output sensitive to the discrepancies in the number of corresponding nodes between the 2 models? And if it is there anything to look out for or control for to make sure things are lining up between cartilage and bone?
Yeah, that's a good question. So in the model we had here for the knee, we had the mesh was finer for the cartilage just because putting it into the COMAK model, we want it was the cartilage mesh that's kind of being used more by the model. So we wanted a finer resolution mesh there. So there were more points on the cartilage. I don't think it matters too much. Possibly the PCA could sort of pay more attention to the surface that has more points, but I don't think it's going to make a huge difference. So it's not something where you need to have the exact same number for all the surfaces. I think as long as it's not wildly different, it's probably not going to cause any issues.
Yep. Another question from Rajan Bora. So you updated the ligament attachments. How did you update the ligament properties at all?
Yeah, so what we did for the ligament properties, so it had set us, I'm trying to remember exactly what it was. I believe the slack length is set, or the reference strain. There's a reference strain that's set. So we kept that the same for all of them, but then we kind of updated what the default position was, where it's getting the slack length from based on that. So it's sort of based on what the sort of optimal settled position is based on that shape, is kind of like, this is the resting position. So from that position, set your slack length. But we didn't actually change the properties aside from that. So we wanted to keep basically everything the same except for the shape.
Okay. Next question from Naomi Frankston. How did you decide on how many segments to use for the model of the ligament?
Yeah, so that was, I was using the model created by Lenhart and the team from University of Wisconsin. So I don't know exactly how they decided how many fibers to add for each ligament, but there's about 6 for most of them, I believe. So I think they just kind of tried to cover the cross-sectional area as best they could.
Yeah.
Something like that. But
Yeah. Next question from Hans Kynes. For your COMAK simulations, do you keep the ligament properties the same? Well, I guess it was related to your earlier question, but did you change the stiffness and
yeah. So
and
in this
yeah,
case, we kept all the ligament properties the same across all of them. Again, just because we were really trying to isolate what's the impact of only changing the shape. But definitely possible that, especially if you're looking at pathology, that certain shapes might be associated with differences in ligament properties and things like that. But here we just kept them the same throughout.
yeah. Right. Next question from Simone Summer. What difficulties do you see when trying to automatically sample bone geometry and ligament/muscle attachment sites from a statistical shape model, i.e. for Monte Carlo simulations and creating a musculoskeletal model from it?
Yeah, okay, that's a good question. So this kind of depends a bit on how you do the correspondence and what your meshes are like. Because when you do the correspondence, you can keep like the deformed mesh, like it's deforming the reference mesh to match your new shape. So you can use that, or you can then try and find what's the closest point to that deformed mesh on your original mesh, which might give you some kind of funny sized triangles in your mesh if you do that. Which then so I guess that can make it worse, but in either case, you might have something where if you're just randomly generating a shape, if you're kind of in something extreme, you might not have a good mesh quality coming out of it, which could affect your simulations after. So that's, I think, would be the biggest challenge, is just verifying that your mesh quality is still good enough to use in the model, which I've found it's usually okay if you're not doing anything too extreme, but it sort of depends on how you prepare the meshes and everything.
Next question from Ariel Sereda. Thanks so much for a great talk. I'm curious if you could recommend current methodologies for modeling elasticity and forces of muscles. Could these be rendered similarly to the bone method you've shown here using a statistical shape model from MRI in specific ranges of motion or positions of the body?
Yeah, so talking about like creating a statistical shape model of muscles, you can definitely use the same approach. The challenge with soft tissues is that they can deform. So you might have like, it might be a different shape depending on the position and things like that. And it can make doing the correspondence a little bit more difficult just because you could have more variation in the shape depending on all those things. So that can be a little bit more challenging, but technically the same approaches can all apply. I know I was talking to somebody who was trying to do a shape model of the Achilles tendon and she was having trouble with the correspondence. But then what I had suggested to her, which apparently worked, is if she scaled the height of the reference mesh first before doing the correspondence, then it made that kind of work out better and she wasn't getting such a kind of funny shape at the end.
Very interesting. Really cool. Let's see. Next question, Seth Kuso. Have you encountered geometries that quote unquote break the musculoskeletal model because the shape is such an outlier? Or if so, how do you approach those cases?
Yeah, so it's definitely possible. I think in this paper, we kept it to like within 3 standard deviations, and that didn't seem to cause any issues. But again, depending on kind of if you're dealing with pathology, you might have more of an issue with that. So like in this model, for example, for the contact, it's only used in the cartilage. So if you had something where the cartilage is getting really small, that could end up causing problems with the model. Or if you're having the kind of bad meshes come out of it, then that can cause problems too. So I don't know what you can really do about it. If you have a shape that breaks your model, it probably depends on what the model is, if there's something you can do to correct for it or not. Some people will kind of take the generated mesh and then do some extra processing on it if needed to make it work with the model. Like maybe you need to crop something off or add a little bit, or I don't know, depending on what you're doing. So there can be like extra processing you could do after to try and make it work, possibly.
Okay. Let's see. Question from Elizabeth Bowman. Could you expand upon how you align the automated coordinate system of the knee to the OpenSim coordinate system? Did you use a rotation matrix, and did you have to export the VTP files in OpenSim and find the alignment between the automated coordinate system and the VTP files?
Yeah, so the method that we followed was there's a paper from Daniel Miranda that has like a procedure for creating coordinate systems for the tibia and femur, and one from Mike Rainbow for the patella. So we used those to basically automatically create the coordinate systems for each bone in our that we had in our data set, and then that kind of defined the coordinates of our generated bones as well. And then basically we just registered the bones in that coordinate systems, like so it's set at 0, 0, 0 in that coordinate system and aligned with that, and then use that mesh for the OpenSim model, so it's directly aligned with whatever those axes are in OpenSim. Trying to think of what the rest of that question was. Is that
Oh,
it?
sorry, I clicked answered on it. So let's see. Yeah, I think it was just did you find alignments to the VTP files was the second part. Like,
Oh
did you
yeah.
use a rotation?
So that is an approach that some people use, is try and like align the new mesh, kind of get it to match with the VTP file from OpenSim. So that's another approach we can use. We just kind of said, okay, if this is our XYZ axis, then put that directly as the OpenSim's XYZ axis. But that's another way you could do it.
Definitely. Okay. Well, unfortunately, we're not going to make it to all of the questions, so we are out of time. But thank you again, Allison and Allison and Aaron. I'm sorry, for a great talk and tutorial, and thank you to everyone for the great questions. So please be sure to click on a link that will appear at the conclusion of the webinar and take our survey. That'll help us improve the webinar series and choose upcoming topics. And this webinar series is supported by 2 grants from the NIH, including the grants that support our Mobilize Center and Restore Center. We're grateful for the support which allows us to run these webinars and other events. And thank you everyone for participating. We hope you continue to stay involved with our centers and that we will see you again in a future webinar. Have a great day.